So thank you so much, team, for applying and for making time to attend this important meeting. Like I mentioned, my name is Namakando Wilfred. I do serve as the Provincial Quality Management Systems focal point person for Central Province. I am a biomedical scientist based at Kabwe Women, Newborn and Children's Hospital, where I also work as a quality officer there. So to begin our meeting, I'll just ask Gomezan to also introduce herself, and then we give you a bit of background to this training, and then we invite Mutale from CIDAS to give us a few remarks.
Thank you so much, Wilfred. My name is Montali Gomezian. I work at Kabwe Central Hospital as a quality officer. Thank you.
Thank you so much, Gomezani. On the technical side, we also have Mr. Chivu, who's just assisting us with Q&A. So maybe Mr. Chivu, also you can introduce yourself, and then we give the floor to CIDAS.
Good evening, everyone. My name is Chibu Felix. I'm a biomedical scientist, and I serve as a deputy quality officer and also a safety officer for the Central Province from Kabwe Women, Newborn and Children's Hospital. Thank you.
Thank you so much, Felix. So as Central Province, we've been trying to make strides towards implementing quality management systems. And through interactions that we've had with people over the last few months, we've discovered that there's actually a knowledge gap among our staff, and probably that would explain the low numbers that we have of labs implementing quality management systems. So to begin, as an initiative, we've begun that we can do virtual trainings and have labs have a starting point in QMS. So for this training, we aimed that we have 40 people trained from Central Province, and we did receive quite a number of applications, and we had to vet, and we ended up selecting 58. And we're hoping that through this meeting, we are going to have a starting point and network within the province. So this meeting is also made possible by CIDAS Trails, and we have Mr. Mutalimbanga from CIDAS to give us a few remarks and encourage us on our journey as we begin. Mr. Mbanga, if you're on call, please can you address us?
Good afternoon, everyone. Thanks very much for the invitation. I think for me, when I heard that you guys were taking the initiative to have a meeting like this or a training like this, it gave me great joy because I have longed, for a day when Zambia and Africa would be ablaze with QMS. So this is really a testament to the fact that we are being proactive and are not waiting for us to be invited to some kind of on-site training for us to gain this knowledge, particularly with the technology that we have now, where we can connect, we can network, even virtually to the extent that we can. The other claim that I have always made is that no one has a monopoly to QMS. It's very, I've been like in the QMS space for quite some time, and sometimes people can act as if QMS belongs to them alone. I don't subscribe to that. I believe that QMS is for everyone. I write about this on my LinkedIn profile, Facebook, especially LinkedIn, where I keep things strictly professional. So QMS, leadership, lab, and everything. And one of the posts I made there was that no one has a monopoly to QMS. It belongs to everyone. So for me, this initiative is great because then it expands the reach of the program, and it also builds leaders that will take up QMS coordination at provincial level or country level, or even at continental level, and even at the world level as well. So congratulations to the organizers, Wilfred Gomeziani, and the team that's behind this. And I wish everybody good luck as the meeting goes on. And if there will be any other support that we can give as siders, we are happy to do that. But also these are kinds of platforms that we are also seeking to build and enhance through the Biomedical Society of Zambia, which belongs to all of you. So we just have to structure it, and efforts are already underway and advanced platforms have already been set. So I think more will be coming. So we are really happy to be part of this, and best wishes. Thanks.
Thank you so much, Mr. Mtale, for those wonderful remarks. So we begin by setting a few rules of our meeting. So for the general chatting, we'll be posting in the chats, but for the questions that the speakers will be responding to, we'll be posting them in the Q&A. Felix will be coordinating the Q&A. Where we can, we'll allow for voice questions. But as you may be aware, this is just an hour, 30 minutes long training, and we have got a lot to cover. So we'll try by all means to strictly follow questions that are posted in the Q&A. I hope that is clear. And then we'll have a brief session at the end of each presentation where these Q&As will be addressed. Thereafter, we'll proceed to the other segment. Should we still have time, we'll look into a practical way of developing an SOP from scratch, and then we'll also discuss the way forward from there. So to begin, Gomezani will take us through the first presentation.
Good afternoon, everyone, and welcome to our first provincial quality management system training, which is Introduction to Quality Management Systems and SOP development. So this is our learning objectives. At the end of this activity, we should be able to define and explain the importance of a quality management system, describe the cost of quality, list the 12 quality system essentials, differentiate national lab certification from accreditation, and identify the different packages of QMS implementation used in our labs.
You want ice cream? It's cold.
May we kindly mute our mics? I'm getting feedback. Wilfred, kindly help me mute people's mics. Thank you. So when we look at our first slide here, it's asking us what QMS is. So a simple definition is a set of coordinated activities to direct and control the operations of the lab with regards to the value of services being offered. So this simply means that all the planned actions we take to make sure our labs work properly and produce results that are useful, reliable, and trusted. So I would like us to break down this first sentence. So if you look at the set of coordinated activities, these are different activities working not in isolation but together. And I would give an example of sample collection, equipment maintenance, IPC testing, and results reporting. So there's no way we can have the other without the other. We can't have results without having a sample. So that's what we mean when we say a set of coordinated activities. The other part that I would like us to break is direct and control operations of the lab. So direct means that we are simply guiding, planning, and giving clear instructions on how work should be done in the lab. So I'll give an example of policies and SOPs, which give us clear instructions on how to perform tests, where we ensure everyone follows the same method as they are conducting a test. So this is where we also define roles and responsibilities. So here we talk about who receives samples, who tests, and who verifies results. Then we can also talk about work plans and schedules. So these simply tells us who's on duty, or rather what tests are being performed and when. So when we talk about directing operations of the lab, this is what we simply means as we define what QMS is. And the other part is controlling lab activities. So this simply means making sure what we have directed is actually happening correctly. So how do we ensure that what we directed is happening correctly? So this is where we bring in aspects of quality control, supervisions and review, and audits and assessments. So QC is simply helping us check that test results are accurate, and it also helps us to simply identify errors before results are issued out. When we talk of supervision and review, this is where we have a second eye checking worksheets, verifying and authorizing results, just to ensure that the test procedures were done correctly. So we're just trying to ensure that whatever we directed is now being done in the right way. So value means quality, and it can either be good or poor. And good quality saves lives, while poor quality has the potential to kill or worsen patient conditions and waste resources. So when we talk of good quality, it simply means we have results that are accurate, reliable, and timely. So when we properly direct and control lab operations, we add value by ensuring results are accurate, timely, and reliable to patient care. So the other aspects that I would want us to look at as we define what QMS is the value of services being offered. So here we are looking at how useful and reliable, as well as impactful the services that we provide as to patient care and public health is. So this is the benefit that patients, clinicians, and health system at large gain from the lab's work. So what makes the lab services valuable or of quality? When we talk of value, what makes our services valuable? So we can talk about accuracy of results. So results that are correct and reliable are supported by QC, EQA, and competent staff. If we have a wrong result, it simply leads to lab services that have no value, even if that result is reported on time. The other factor that can make our lab services of quality is timeliness. So here we are talking about results that are available when needed for clinical decisions. So here we simply talk about our turnaround times. What we should know is that a late result reduces clinical usefulness. The other factor is reliability. So when we talk about results that are reliable, we mean results that are dependable and trustworthy. So the question that we should keep on asking ourselves is, do clinicians have confidence in the results that we issue out? And a typical example that I can give when we're talking about reliability is, if a patient comes with an HB of 10 today and 14 of, if a patient comes with an HB of 10 grams per deciliter today and 14 grams per deciliter tomorrow without treatment, clinicians will question the reliability of our results. And we should ensure that clinicians have confidence in our lab results. then they can safely make decisions based on our results, and this increases the value of lab services. So QMS is existing to maximize the value of lab services by simply ensuring results are accurate, timely, reliable, and safe. So we can move to cost of quality. So what do we mean when we say cost of quality? So this is simply the total cost the lab incurs to ensure quality and the cost that results when quality fails. So when we look at our slide, the total cost of quality is simply cost of good quality plus cost of poor quality. So in other words, it is the money we spend to do things right and the money we lose when things go wrong. So when we look at cost of good quality, what is it? So this is money spent to achieve quality services. So in short, these are planned and necessary costs to prevent errors and ensure reliable results. So when we talk of cost of good quality, we have two factors that can fall in cost of good quality. So we have prevention costs as well as appraisal costs. When we talk of prevention costs, these are costs that the lab incurs to stop errors before they happen. So as we start implementing our quality management system, we'll have to incur certain costs so that we ensure that errors do not happen. And these include staff training, competence, SOP development, as well as equipment maintenance and calibration. And when we talk of appraisal costs, these are costs the lab incurs to detect errors early. So these may include the internal QCs that we usually run, external quality assessments, audits, and supervisions. So the other cost of quality is cost of poor quality. So when we talk of cost of poor quality, these are unplanned costs due to mistakes and failures. So under cost of poor quality, we also have two factors. So the first one is the internal failure costs, and the second one is the external failure costs. So when we talk of
Don't get conscious.
Sorry, I'm getting feedback. Wilfred, can you help me to? Thank you. So when we talk of internal failure costs, we are talking about failures that are detected and controlled and corrected inside the lab before results are issued out. So we notice them and then we correct them inside the lab before results are issued to the clinician. And these include repeat tests, which can be due to maybe untrained or incompetent staff or unmaintained equipment, which frequently gives us errors. In the other one, and all this will lead to reagent wastage. Then the other cost of poor quality is external failure costs. So these failures are detected outside the lab. So this is where the result is issued out, and then maybe a clinician complains, then that's when you notice to say, oh, there was an error here. So these are failures detected outside the lab after results are issued out. And these may lead to incorrect patient treatment, complaints, loss of clinician trust, or legal or reputational damage. So this is simply just highlighting how lab errors cost us. So the errors cost us in time, the time that we spend repeating tests, the energy that we spend running the same sample due to errors, the money that we spend when we waste reagents, patient outcome, and lab's reputation. So this graph is simply illustrating cost of quality. So when you look at the horizontal axis, which is the x-axis, this is simply talking about the level of quality. And when you look at the y-axis, it's talking about the cost of quality. So when you look at the x-axis, this is talking about quality levels. And if you look at the left side here, it shows low quality, and to the right it shows
Bring the
high
remote.
quality.
Where are the remote?
So when we move from the left to the right, it's showing us that we are having better systems in place, better control measures, and fewer errors at this point, because we are moving from low quality to high quality. And if you look at the y-axis where we have quality costs, so we have low costs at the bottom and high costs on top. So this simply represents money, time, resources, and efforts that we put in place to ensure quality. So when you look at this the blue curve, the failure costs, it's simply telling us that when quality is low, failure costs are high. So in our labs, if our quality levels are low, the failure costs are high. We'll end up having errors, repeating tests, wasting reagents, and so on and so forth. And one thing that we should know is from this graph, it's simply telling us to say poor quality is expensive, because if we have no quality at all, we'll have high failure costs. Then when we look at the blue curve on our right, it's simply showing us to say, as we invest more in quality, prevention and appraisal costs begin to increase. So this includes training, SOP development, QC and EQA, as well as equipment maintenance and audits. And then when we look at the red curve, the total quality costs, this is simply telling us to say, this is the sum of costs of good quality and the cost of poor quality. So at low quality, total cost is high. So when we look at low quality here, the total quality cost is high. And we have many failures and cost of poor quality, which is also high. And then when we look at, so this simply means to say at a very high quality, total cost can also increase. When we look to our right here, we have high total quality costs as we increase our quality levels. So, but somewhere in the middle here, it's simply showing to say the total cost is low. So when we look at this green dotted line, it's simply showing us to say this is the optimal quality level where the total quality cost is lowest. So at this point, this is where we have failures well coordinated, we have prevention costs that are reasonable, we have resources that are used efficiently, and we have patient care, which is protected. So if we don't run QC, maybe we are trying to save on money today, we'll end up spending more tomorrow, repeating tests and correcting results. So as we take a step and start implementing QMS in our labs, our goal shouldn't necessarily be perfection at any cost, but optimal quality where everything is balanced. So as we talk of good quality, how can we achieve the same good quality? So quality does not mean doing things our own way. It means doing things in an acceptable, agreed, and proven way. So to achieve good quality, the lab needs an acceptable way of doing things. So an acceptable way of doing things is not personal preference, but it is based on evidence, research, and expert agreement. So this simply helps us to ensure consistency across labs. So this is where now the ISO comes in. So the International Organization for Standards, which is the ISO, develops international standards that define how quality should be achieved. So the ISO tells us what must be in place for quality and not necessarily how to do every step. So it will just guide us to say, for us to achieve good quality, we need this, that, and that. But how we are going to go about implementing those things, it doesn't restrict us. So guidelines are also drawn from the standards to make the implementation process easier, because sometimes standards can be complex. So guidelines are developed to help labs implement them more easily. So this is where we have the National QMS Certification Guideline, and others that are implementing the ISO standards are undergoing accreditation. Okay, so this is just simply talking about the ISO. So it is derived from the Greek word isos, meaning equal. So like we said, for us to achieve these things, we are supposed to be doing things in an acceptable, agreed, and proven way. So if two labs are meeting the requirements of the same standard, then they are equal. If a lab here in Zambia is, I'll give an example of the ISO. If a lab here in Zambia is meeting the requirements of the ISO 15189, and another lab maybe in Asia is also implementing the ISO 15189 and meeting its requirements, those labs can be called to be equal because they are meeting the standards of the same, they are meeting the requirements of the same standard. So ISO is an organization with representation from 137 countries and makes discipline self-specific standards. So that's just an example of an ISO standard, which is 15189 2022 for medical labs. And we also have ISO 15190 for safety in medical labs, among other standards that we have. So are we meeting the standard? And how do we know that we are meeting the standard? So an independent body provides formal recognition that the lab is competent to carry out specific activities in accordance with a given standard, or that it has a management system that complies with a standard. So we have independent bodies that recognize a lab for being competent or meeting the requirements of the standard. So the well-known independent body that we have in Zambia is SADCAS, that accrediting labs for them implementing the ISO 15189. So formal recognition of technical competence is known as accreditation. So formal recognition of conformity, compliance with a management system standard is known as certification. So we have labs that are in the accreditation process, and we also have labs that are in the certification process. So this slide is talking about the importance of QMS. So why is QMS important? So QMS simply ensures consistent and reliable results. So once we start implementing QMS, we won't have each individual testing samples in their own way. So it brings about consistency. So it will guide us to say, if I'm working on the Cobas C111, what am I supposed to do? It will guide us to say you're supposed to follow these steps. If I'm working on the Pentra C200, I'll follow these steps. And if another person comes through and is working on the same Cobas C200, we're supposed to follow the same steps. So it also improves customer or patient satisfaction. So our customers will be satisfied with the services that we offer because they'll have confidence in us to say the results that we're issuing out are reliable results and accurate. So QMS also reduces error and wastage. So once we start implementing QMS, we'll know that we'll have to do equipment maintenance. Maybe those are some of the procedures that we skip. We'll know that we're supposed to run controls to help us reduce on errors. The other one is supports continuous improvement. So QMS is all about continuous improvement. You can't say once you start implementing QMS, then you've reached, you are there, you won't be doing anything else. But it's all about improving the process, making sure that everything is in place, and better staff competence and confidence, as well as required for accreditation and certification, because we can't be accredited or certified if we are not implementing QMS, because definitely they will need to see some form of QMS implementation for us to be accredited or certified. Okay, so and now to the 12 quality system essentials after having looked at the cost of quality and the introduction to QMS. So the 12 quality system essentials are the foundation of an effective quality management system. So they simply ensure consistency, accuracy, compliance, and continuous improvement, like we talked about. So these are the 12 quality system essentials as described by CLSI. So our first quality essential is organization and leadership. So what do we mean when we are talking about organization and leadership? So we are talking about leadership and management commitment to quality. We're talking about quality policies and quality objectives, organizational structure, defined roles and responsibilities, as well as authority. So quality starts with leadership and clear accountability. We need to know individual responsibilities for us to implement quality management system. The next essential is personnel. So here we talk about staff qualifications and job descriptions. We have people working in the labs, but do they have job descriptions? Do they know what they are supposed to do? Are they qualified people that are supposed to work on patient samples? So we also talk about training and orientation programs. Are the people working in the labs trained? Are they oriented? Yes, we go to school and we learn our medical sciences, but when we come to the lab, do we get trained on how to operate equipment? Do we get trained on how to do gram stain? We also talk about competence assessment and evaluation. After a staff is trained, we have to deem those people competent. So do we deem our personnel competent to carry out procedures that we authorize them to do? And also, we also talk about continuing professional development. So our key message under personnel is that competent staff produce reliable and accurate results. If a staff is not competent, if a staff is not trained, how do we expect them to issue out reliable and accurate results? So we can also look at the next 12 quality essential, which is equipment. So when we look at equipment, we talk about equipment selection and installation. How do we acquire this equipment? How do we select which equipment best suits our service delivery? And how do we install this equipment upon receipt? It also talks about validation, calibration, and maintenance. How are we going to validate or rather verify our equipment after we receive them? And how our calibration is going to be done? And how is maintenance going to be done on these equipments? We also talk about equipment operation and user training. We need to train our staff on how to use the equipment. Then we can also look at equipment records and logs. So a well-maintained equipment ensures reliable test results. Our next 12 quality essential is supplier and inventory. So here we're talking about supplier selection and approval. Once we start implementing QMS, we don't just buy reagents from whoever is selling reagents. We need to approve suppliers. So we need to select and approve those suppliers. We'll also talk about procurement procedures. How do we procure a reagent or a consumable? We also talk about inventory management. storage and stock monitoring. We talk about acceptance testing. If we procure a reagent, do we directly start using that reagent, or we have to do acceptance testing where we test to check if that reagent is fit for use? Then we also have record management concerning reagents and consumables. So our key message under purchasing and inventory is quality materials are essential for quality testing. So we have to ensure that whatever materials we procured are of quality, because expired reagents can lead to incorrect results. Our next quality essential is process management. So here we talk about standard operating procedures. How do we control our processes? We have pre-examination processes, examination processes, as well as post-examination processes. So how do we control them? We talk about standard operating procedures that guide us on how we conduct tests. We talk about internal quality control, EQA. We talk about method validation as well as verification. Are we verifying our methods? Yes, the manufacturer has told us that if we use a gram stain kit, we'll be able to get a gram positive or a gram negative bacteria organism. But if we receive those reagents, are we trying to verify to say what the manufacturer has said is what the kit is giving us? So a process must be controlled to ensure accuracy and consistency. The next one is information management. So when we talk about information management, we are talking about reporting and release of results. How is this going to be done? It talks about traceability of methods being used. If we issue out a report or release the results, do we indicate to say this is the method that we used? How do we trace back to which method was used? If you have two analyzers in a chemistry section, do we indicate to say this result was processed using, this sample was processed using maybe the Cobalt C111 or the Pentra C200, so that in case we want to follow back to how the QCs came out, if we want to check if that result is accurate, how do we trace back to which method was used? We talk about information systems, be it electronic or paper-based. We talk about data protection. How do we protect our data? Is everyone accessing our data? Is everyone accessing patient information? Then we also talk about authorities for information management tasks such as access and amendment. Is everyone going to be able to access our information? Any clinician that comes through the lab, is this person going to access our information? Maybe if we're using paper-based and maybe a clinician comes through our lab, are they going to have access to our books? Or if we're using electronic system, are they going to be able to have access to whatever system we're using? Also, we talk about amendments. Who's authorized to amend results? Is it everyone who is authorized to just maybe receive samples, authorize results, amend reports, and all those things? So information management guides us on how we go about all those things. We talk about documents and records. So here we talk about legal entity, policies and objectives, documents control, as well as records control. So when we talk of legal entity, we are talking about a lab being, is it a private or a government lab? And is it maybe, is a lab under an institution, maybe a hospital, or it's a standalone lab? All those things we're supposed to define. Then we talk about our policy. What is our policy? What are our objectives? And how are we going to be controlling our documents as well as records? So our key take-home message on this one is, if it is not documented, it did not happen. our next one is non-conforming event management. So here we are talking about identifying NCs, classifying NCs, sources of NCs, cause, immediate, and corrective actions. So as a lab, we normally come across non-conformities. So we need to have guidelines on how we are going to deal with those NCs. How do we identify the non-conformities? Because if we don't define how we're going to go about all those things, we may be thinking that, no, my lab is perfect, we don't have any non-conformities, but maybe it's because we didn't define how we're going to identify non-conformities, or rather how we are going to classify them, and where do we look out for the sources of, where do we look out for these non-conformities? So we define the sources. Is it internal audit? Is it risk? Is it, so whichever sources that we define for, as long as at least we have a guide on how we are going to identify NCs. Then the next one is assessments. So here we talk about internal audits, external assessments, and risk management, as well as quality indicators. So for us to know that we are implementing quality and we are moving in the right directions, we need to audit our systems. So we talk about internal audits or external audits, as well as risk management. It helps us to know in which directions we are heading, as well as the quality indicators. So these assessments, they will show us whether the QMS is working effectively or not. And we'll need to implement further action so that we move in the right directions. The next one is continual improvement. So this is simply helping us identify, implement, and communicate opportunities for improvement. So like I said, when we start implementing QMS, it doesn't mean our lab is perfect, but we continually improve our system. It's all about, QMS is all about continuous processes. So we use data to improve our system. We can, maybe let's say, for example, we have complaints towards a certain area. We can use that data to say, no, we are having a lot of complaints in this area, so we need to improve in this aspect. So we implement actions so that we improve our system. The second last is customer focus. So here we are talking about advisory services, handling of complaints and feedback, service agreements, lab information for patients and users, as well as utilization of customer feedback. So when we talk of advisory services, these are simply maybe meetings that we can be having with our lab users to advise them on the services that we offer as a lab. Because we may be thinking our lab users know to say we have such and such a test, but in the actual sense, they don't know. Or maybe we can be thinking our lab users, they are aware of how we operate as a lab, maybe how we receive, how they can lodge in their complaints and all those things. So these advisory services are held so that we inform our lab users on how the lab operates. Here we also talk about handling of complaints and feedback. So as a lab, our customers are the lab users. So we also talk about how we are going to be, definitely there'll be complaints. We can't say, me, my lab is perfect, we don't have complaints. So we need to have guide on how we are going to handle complaints and feedback. Also, how we are going to deal with service agreements, as well as information for patients and users. We need to provide them with information on how the lab operates, how the services that it provides. And then the last one is facilities and safety. So here we talk about safe working environment, specimen transport, biosecurity, access control, containment, waste management, fire safety, chemical and ergonomics. So we, as a safe working environment, supports quality services. If we don't have a safe working environment, definitely we can't have quality services. So as we implement QMS, we also need to factor in the aspects of our facilities and safety. So as I conclude the first part of today's training, our take-home message is that implementing quality management does not guarantee an error-free lab, but it detects errors that may occur and prevents them from recurring. So it doesn't mean the labs that are implementing QMS are error-free. No, even if you were to go to a lab that's implementing QMS, you yourself, you are also able to notice errors. So it doesn't mean that it's an error-free lab. So this slide is simply just giving us a clue on the QMS programs that are available. So we have accreditation, national certification, strengthening lab management towards accreditation center, as well as low-level QMS. Thank you so much. Mr. Namakando, over to you.
Thank you so much for that clear explanation, Gomez. Team, if you have any questions, we can post them in the Q&A. So this meeting has been made possible with support also from the Central Province Health Office through Dr. Grant Nombwende, who's also on call. Dr. Grant, please, you may grace us with a few remarks.
Yeah, thank you so much, Wilfred. And may I just welcome everyone on call. I can see the meeting is well attended. I've counted so far 44 people on call. This simply shows how people appreciate QMS and how much they regard it to be important. I also wish just to thank you, Gomeliani, and Wilfred for this initiative. Learning is not only by sitting in class and seeing the lecturer or the tutor physically. With technology now, you can from the comfort of your home, while traveling, or whatever you are doing. So in short, it has allowed us to multitask. So you can do quite a number of things, with virtual training. This, I believe, is also cost effective because there's no accommodation to be paid. If there's only subscription, probably just for bundles and alike. So let's take this opportunity and we pay attention to it and hoping that all those that are on call now will run all the way to the end of the program that you just started. I know virtual training has its own challenges at times, especially that I've observed it's just too quiet. I'm just hoping that everyone is really listening and not just logged in and away, from the presenters. As a president, we're really happy, that we have our staff dedicated to quality management systems, and that you really take upon it. You've seen that actually for your results to be credible, to inculcate the sense of confidence in our clients, the clinicians, and the rest that is our results of the product. You need to give them a good quality product, and this can only be done through a quality management system. Not take a lot of your time, but just to thank all of you, the participants, for being part of this, and also the trainers. With the reporting, this was already reported in the management meeting that there's this initiative and it will be taking place, and we did. I'm happy that it has materialized. And so, with these few words, without taking much of your time, I just wish to thank all of you. And please, let's proceed and enjoy the next session. Thank you so much.
Thank you so much, Doc, for those remarks. The team that we have here is actually coming from all the 11 districts, and Kabwe is the one that has the highest number. We received quite a number of responses towards this training. However, we want this training to have a follow-up, so we wanted to minimize the number of people that we invited, hence the number that you are seeing here. But we did have more than the number that we are seeing here that had applied. So we hope we build connections from here, and we get to interact and push the agenda of QMS within our labs. Thank you so much, Doc, again for those remarks. Mr. Chiwu, do we have any questions in the Q&A? Please guide us through that session.
All right. Yeah. So the first question is coming from Madam Karen Wadia. She's just asking Madam Gomeziani if she can go through the last slide, if she can explain more. Then the second question is, are these presentations going to be made public to the participants? Then the third question is coming from Mr. Nevi Tembo, which says, hello team, thank you for the great initiative of strengthening quality management system, as it is the core of laboratory services. I want to find out at which point does SLAMTA and SLAMTA fall in the system, whether at initial phase or implementation, or whether at initial phase of implementation or where exactly. Just a thought, considering that it may be a bit difficult to employ all the 12 quality essentials initially. That's the last question coming from Mr. Nevitembo.
Okay, so I think one is coming from Mr. Pole Pasco, and it reads, regarding the different types of QMS programs, how do you determine which type is appropriate for a particular lab? And secondly, if a particular lab needs to start working towards accreditation or certification, what is the starting point? So I think those are the questions that we have so far. Well, with the other question regarding if these notes are going to be made publicly available, the answer is yes. Immediately after this meeting, via that very thread of emails that you received your invitation, these notes are going to come there. So just to begin to answer the question that came from Navy and the question also that came from Mr. Pole, we'll begin by saying quality management systems, really, the aim is for us to improve patient well-being. And some of these programs that have been put ahead of us are really just to help us reach that particular objective. So currently as a nation, we have these 4 here that are showing on screen. One of them is called the accreditation process. The other one is called the national certification process. The other one is called the strengthened laboratory management towards accreditation, which is also known as SLAMTA. And then we do have low-level quality management systems. As per policy or as per objective by the government, we know that we need to take services as close as possible, quality services as close as possible to the community. So as we are beginning 2026, we do not expect any lab not to be implementing any form of QMS, being a health post all the way to the tertiary institutions. So now the package that a lab gets to choose to implement depends on the resources and the infrastructure that the lab particularly has at that point. So for labs that are at health posts and do not have equipment, they do not have sufficient infrastructure, what they are going to be subjected to is the low-level quality management systems. So this will ensure that especially their process requirements are up to a standard, even as other things such as infrastructure, access control, storage of commodities is not yet well organized at that particular institution. So for those health posts especially that don't have space, don't have personnel, don't have the equipment, will do a low-level quality management system package. So this will only look at EQA, to look at your competences, to look at the IQCs, but it will not go on to look at the biosafety, biosecurity issues. It will not look into risk. So that's about the low level. Then when we come to strengthening laboratories towards laboratory management towards accreditation, which is SLAMTA, here the goal is for those labs that have not implemented before. So the concepts of quality management systems are new to them and they want to begin. So they begin through a process which is known as strengthening laboratory management towards accreditation. So here they get to undergo a series of trainings, and then those trainings come with tasks. As they are completing those tasks, they are becoming familiar with how to implement. So this particular one is star-based kind of program. So you have labs rated from 0 star to 5 star, depending on how they perform on a checklist. So I'll combine this with Mr. Nevi's question, who was asking about the SLIPTA and the SLAMTA. Where do they really fall? So SLIPTA is a checklist that is used in SLAMTA to check the lab's process, to check the level at which they are moving at. So the star ratings will range from 0 to 5 star. The assumption is that the moment you reach to 5 star, it means you've become familiar with the processes of quality management systems and you are able to apply for accreditation. Now, when we go to the National Certification Program, so this program was designed by the Ministry of Health Zambia through the Quality Assurance Coordinating Unit, particularly to be implemented by laboratories that are, one, beginning quality management systems as well. Number two, it also prepares them for accreditation. So this is also based on the same standards as accreditation is based on, just that a few things have been removed since these people that are implementing are just beginning their quality management systems. So also, this is also star-based. It ranges also from 0 star to 5 star. The accreditation program itself, this is where now an international body such as SADCAS would come to your facility and then give you international recognition for quality and competence for your lab with specific claims to a particular test. So if you're doing well in your RPRs, then they'll accredit you in RPR. So to answer the question now of where do you begin? So as a lab, we need to look at how is your infrastructure? How are your staffing levels? How are your resources? Do you have money for training? If you do have money for training, you do have money for a number of programs that need to be fulfilled within the requirements of the standard. then there is no harm in jumping straight into accreditation. But if you want to take baby steps and see that processes are built and to include issues of staff commitments, staff attitudes are handled properly, then you may begin either with a national certification or with a SLAMTA program as you are beginning to build those 12 quality essentials, because the SLAMTA program is structured in a way that you're going to build the 12 quality essentials one by one as you are building your quality management systems. I hope that answers the questions.
And just to add on, Wilfred, from what you've said, so as we think of QMS implementation, we should think of it as a journey. It's not something that we are going to achieve overnight, but it's something that we'll achieve as we go on. Like you said, baby steps. So maybe I got your concern, and definitely somehow we can't manage to implement all the 12 quality essentials at once, but it's a journey, and one by one, they are building blocks, not until we achieve what we are working towards.
Okay, so for the second session, we're looking at documentation, which is also one of the key starting points. By the end of this activity, you should be able to define and differentiate documents from records, to describe the hierarchy of documents, to discuss SOPs, what they are not, and their roles in the quality management system. Also, you should be able to explain the document development process. A document is any written item that contains information or data in an accessible form and can serve as a means to gather objective evidence. So here, let's break it down first. We are saying that the document is supposed to be written and accessible. So those are the first things that we consider when we say a lab has got documentation. It shouldn't be verbal. If it's written down, it should be easily accessible. And then it can also be used to gather evidence of activities that are being performed. On the other hand, a record is information created and maintained as objective evidence of activities performed, discussed, or as proof of any transaction that took place. So here we are talking about you keeping evidence of the things that are happening within the lab. It can be a test that you performed. There should be evidence that this test was performed by who, approved by who, released when. There should be that trail. Should you have a meeting within the lab, you're discussing matters, there should be evidence that such meetings took place. All the transactions that a particular lab makes, there should be proof, and that proof is what is known as records. So now, documents, the written accounts, need updating whenever we learn new information. So if you knew a way to do Gramston today, and then tomorrow researchers find that there's a better way, and they validate it and publish to say that the way we were doing Gramston has been outdated, the newer way is this way. Whatever document that you had within your lab that was talking about Gramston will now need to be updated with that new information. Now, the same documents can also be used to gather objective evidence, like we said before. An example is a temperature log. When a temperature log is blank, you have not used it, it's a document. And if you find a better way of documenting temperature, you can update it to that better way. But once you fill it in, it becomes permanent. You can't change it. And that's what becomes a record. So every other document, once you fill it in, it becomes a record, because now that is permanent, you can't change. It's objective evidence of what took place. Let's look at the documentation hierarchy. We now know from the previous slide that we only have two types in our system, two types of documentation. The first one being documents and the other one being records. So the types of documents that you can have in the lab, the highest level of documents is known as policies. So these policies are here to give us an intention of what the lab is about. So to begin with, it just describes what happens in this lab and why does it happen. Examples of policies are manuals that we have in our lab. I'll give you an example of the quality manual. So in the quality manual, would describe what the intentions of the lab are. Would describe and say, this lab has got intentions of producing accurate, reliable, and timely results. The reason why that is happening is to ensure that we protect patient well-being, and we see to it that these results that we're issuing out are the correct ones and speaking to the conditions that these patients have. So we'll document all of those statements of our intentions into a policy document, such as a quality manual or a safety manual. And that becomes what guides the rest of our documentation. From that policy, we can have processes. These are sequence of events to show us how those intentions are going to be implemented within our lab. And then from there, we can have detailed instructions of how each and every process is going to be achieved. So those documents now are going to be called procedures, the ones that show us the actual steps of how the sequences of our intentions are going to be fulfilled. But sometimes these procedures become too long for us to be referring to them. You'd find a procedure is 12, 15, 30 pages long, and for you to flip and find information might not be easy. So we extract from the procedure or the process or the policy to make an abbreviated instruction. Such an abbreviated instruction is going to be called a job aid. It's just a reminder of how to do a particular task. It's not the procedure itself, it's just an abbreviated instruction from the procedure itself. So we have seen a number of labs when you visit and you ask for procedures and they give you a job aid. A job aid and a procedure are two different things. A procedure is a detailed instruction and a job aid is an abbreviated instruction, just showing us reminders of how certain steps are taken. And then from there, as we are performing these activities, we need to have a tool that we are going to use to document that the activities that we've done at policy level, at process level, at procedure level really happened. So those tools are called forms. We're now going to use them, control them, and record our objective evidence on those particular forms. Once those forms are filled in, they now become what is known as records, as that is now the documentation of what has been done by the lab. So for this particular meeting, we'll not look at policies and processes. We'll look at procedures. Policies and processes, we'll discuss them in another meeting because they are quite lengthy on their own. It's a topic on their own. So we'll discuss procedures here. So procedures in our lab are the ones that are commonly known as standard operating procedures, or SOPs. By definition, an SOP is a written step-by-step instruction detailing how a specific task must be performed the same way, correctly, safely, every time, to support reliable, valid, and reproducible outcomes. If we break down this term, we are looking at something that is written. So as labs, we should strive that all our procedures are written. Not when we visit a lab or when our lab gets visited, we do not have anything that is written, but we claim that we do have SOPs. People may know, maybe you discuss through a meeting, people may know how to do certain things, but if it's not written, then you do not have the SOP. Then the second one is that it should be step-by-step instruction, detailing how a specific task must be performed. So it should be step-by-step, and the task that you're describing should be known. It shouldn't be general. It should be known. The reason why we are putting this in writing and step-by-step is that we want to ensure that when a lab personnel A performs a test, they perform it the same way as personnel B. And that way that they are performing it, you might be performing it the same way, but it's a wrong way. So an SOP will ensure that the way you are performing it is correct and is safe also for the person that is you doing that particular task. There are two types of SOPs that you're going to find in a lab setting. One of them is known as management SOPs. These SOPs are going to guide laboratory activities. And then we do have technical SOPs that are going to be used for testing activities. As you may be aware, labs do have two types of activities that they perform. Labs do have two activities that they perform. One of those is to test a sample and issue out a result. On the other hand, we do have also activities that are non-testing. My colleague Gomezani was talking about advisory services where we offer advice to our clinicians on interpretation of results, on what tubes should be used to collect a particular sample. Those are other services. Those are not testing services. So those services are going to be managed by management SOPs. She talked about during personnel, among the 12 quality essentials, she talked about when staff come from the universities, we assume that they know it all and then they are sent to the lab. But the universities did not have the equipment that they're going to find in the lab. So as they are coming into the lab, they need to be trained, they need to be oriented, they need to be trained, competence assessed. All those activities are grouped into what is known as management activities. And you need to have procedures or SOPs that are going to manage those aspects. Okay. So now let's look at what an SOP is not. So guidelines and information from product instructions, e.g. package inserts, user manuals, and user training guides are not SOPs. Would find a lot of laboratories when visited by assessors would present guidelines for malaria and present it when they're asked, do you have an SOP for malaria? And then they take out a guideline from national level and issue to the assessor to say this is the SOP that we have. So SOPs and package inserts, user manuals, all of those are not considered SOPs. Instead, those can be incorporated into the SOP by reference. So you take out whatever information that you need from those reference materials, and then you put into your specific SOP that is going to guide processes within your lab. you'd find not everything in a user manual or in a training guide applies to your lab. There are certain things that are modified in your lab. You would find your centrifuge does not open from the open button you've modified from the back. So all of those things now need to reflect in your own SOP. Textbooks as well, research papers, posters and flyers, all of those are not SOPs. They are reference material that you can use to collect your information that you're going to put into your SOP. So if it does not describe the full steps that are being carried out by your laboratory for a specific task, then it's not an SOP. An SOP should be able to guide you from the point that you receive a sample to the time that you are issuing out a result. So that process should be well documented in your particular SOP. Now, what is the role of those SOPs in our quality management systems? So those are the ones that link the QMS theory that Gomez just took us through to our daily practice. So now what are you going to say about personnel in your own laboratory? You put it now in writing to say for this lab, personnel are going to be treated in ABCD. When we receive them, they're going to be oriented. Orientation is going to be done by this one. This is the material that is going to be covered during orientation. This is where you're going to document that, and you go on to the end until the person is deemed competent. When it comes to a lab test, it will be when you receive a sample, here's where you're going to register it. You're going to register it in this way. It will go to this bench. It will be tested by people that have this authority. It will be approved by this one and all of that. So it now connects what those essentials are and what your lab is doing. So the first role is that it helps us in standardize the processes. If we do not have an SOP, everyone will be doing what they want to do, whichever way they will be doing it, as long as they get the job done. It also helps you to support training and competence assessment, because if you're training someone, there should be reference where you're training. There should be reference that you're going to base your training on. Even in schools, they have a curriculum in which the lecturers are going to use to train you. And then at the end of the training, there should be a criteria in which they are going to use to assess you. So these SOPs now are going to help us, one, train, and then the same ones are the ones that are going to be used on people as they are being observed for competence. It also helps us in aiding risk management and error reduction. So we'll dive more into risk management as we are beginning these trainings. It's one of the trainings that we are going to do. But if you have something written down, it means the chances of you making a mistake have been minimized. It also helps us support continuity of operations. Many of the times we've heard this one has been transferred, this one is sick. If you do not have processes written down, it becomes very difficult for other people to take over. You would find that the hard work that you've put in as a lab, the moment one person changes, if you didn't write things down, all of that hard work goes to waste. It also helps us to prepare for audits and inspections, making sure that we are compliant with the standards and the regulations. I remember Gomezan in one of our first slides, he mentioned that we need to find a correct way or an agreed way of doing things if we are to implement standards. So what Lab A is doing should be the same as what Lab B is doing. So if you have some things written down, then it helps you be compliant with such standards. And very important, it helps us make sure that the processes that we've used to do this testing leads to us producing correct results. So SOPs are the ones that make a functional QMS. So for the management SOPs, there's a link here that we've shared in the notes. The Ministry of Health has drafted management SOPs for us that we can just customize to what happens within our settings. So the link is within the notes. The notes are going to be shared just after this meeting. We can go to that link, download, and then start customizing. Myself, Komezani, and the other mentors that we're going to make available to you are also available to help you where you need help in understanding those particular templates for management SOPs. Please just download, do not edit from that particular link. Just download and then edit offline. Next slide, Gomez. Okay, so let's dive a little bit more about technical SOPs. We've already established that technical SOPs are supposed to be step-by-step instructions for running patient samples from when the sample has been received to when it is reported. They should give specific instructions for what should be done and how it should be done at each stage. Those particular SOPs may also refer to other documents. So if you can't compress a particular process that is in a user manual, you can refer to it specifically and say, for this particular action, you can go to a user manual on page so, and then you look at information that is talking about this. Please, next slide. So let's look at what the standard says about development of technical procedures. So the standard says that SOPs are supposed to be documented in a language understood by staff. Of course, this is not just talking about you putting it in English. It means the English that you're going to use also must be understood by the people that you are making the SOP for. Then those SOPs should ensure validity of results to include, where applicable, the ways to detect trends and shifts. So when they say validity of results, they're just saying quality control checks. The SOPs should include procedures for quality control, and where applicable, for example, for our quantitative tests, we need to have means in which we are going to detect trends and shifts. For those that have been implementing or those that are a bit familiar with QMS, when it comes to trends and shifts, these are detected by plotting your control values, and then you subject them to some rules and see where the shifts are and the trends are in your quality control so that you can take appropriate action. Traditionally, people have been using a reference range that is given by the manufacturer. Let's say HB, they say the reference range for normal is 10 to 12, and then you run your QC, and then if it's within that range, you assume that your QC has passed. But you haven't analyzed your data further to see the trends and the shifts. That's why they say you need to plot and do your Levy-Jennings and see where the trends and shifts are. So good SOPs, according to the standard, should allow for such things to be documented within, and people should follow through. Then staff should also understand and follow those SOPs, meaning there should be evidence. When you do an SOP, there should be evidence that your people have understood, and there should be evidence that they are following those SOPs. And the SOPs are not supposed to be placed in cupboards or in archive rooms. These are supposed to be available at the point of use where these activities are being conducted. And they're supposed to be document controlled. Now, what do we mean by document controlled? We mean that they need to be approved for use. Their versions should be known. If you make an SOP today and then you update it tomorrow, the one that you made today should be known as version 1. The one that you make tomorrow should be version 2. And all of them should be tracked to see where they are, which one is in use. They need to be reviewed and updated with time. As we know, trends are changing, technology is changing. So as things are changing, we need to be updating our SOPs also. We shouldn't have an SOP that was made back in 2000 still being used today. Things have changed. When an SOP has been changed and a new one is in effect, the old one is known as an obsolete version, and it must be removed from the system. Now, very important, all of those SOPs that we're talking about need to be linked to the management system. My colleague earlier spoke to say these are coordinated activities. So those need to be linked together. And a common way of linking them is assigning them numbers so that you can refer to them even in other SOPs. So I would say maybe for preparation of a cell suspension, refer to procedure number 5. For a process about this, refer to procedure number 8, so that you know how your procedures are linking to one another. Next slide, please. Okay, so now the big question is who writes these SOPs in our labs and who should approve them once they are written? The simple answer is that it's only staff that have competence. By competence, we mean they have the theory on the topic that is being discussed, and they also know how to do it. So those are the people that should write SOPs. It shouldn't be the lab head, the quality officer, or any other position if they do not have these qualities that we've talked about, the theoretical knowledge and the technical skills. So those two, if a person has, then you can give them to write an SOP. In a majority of our procedures today, especially the newer methods, when the vendor comes on site to install, they usually do an on-site training, and those are the people that they live with the theory and the technical know-how. So subsequently, those are the people that should be able to write SOPs for the lab. For the odd staff, we know our strengths. Even within ourselves, we know that this one is better than me in this area. Let's not have so much ego. Let's give room to such people that we know actually know more than us, to be the ones that do these SOPs and then train us on those SOPs. Okay, let's look at some of the writing principles for these SOPs, especially the technical ones. So the first one is that we need to use active voice. An example has been set here where we say centrifuge the sample is an active voice. When we say the sample should be centrifuged, we are using a passive voice. So SOPs should use an active voice to give clear instructions of an action that needs to be taken. Then they need to be specific. An example is when you're centrifuging and you're writing an instruction in the SOP about centrifuging, you need to say centrifuge a sample at 3,000 revolutions per minute for 10 minutes. Do not just say spin the sample and it ends there. It leaves room for people to spin at whatever speed and at whatever time. Also, use simple language. An example is mix. We know we are highly educated. Others would want to use the language homogenized by agitation. But really what they mean is just mix a sample. So please make it just as simple and clear as that. Then avoid writing things in paragraph form. Write things in a stepwise form. Say step 1, add 1 drop of this. Step 2, remove this and add to that. Step 3, it should be in those clear steps. And then they should be factual, concise, and clear. What we mean by factual is that whatever words that you put in the SOP should be based on evidence that is available to be checked. So get a national SOP and then tailor it to your own. Use the national SOP as reference of the facts that you've put into your own. Should you use a study, get that study and have it as reference to back your claims that you have in the SOP. If we can't find where you got the information that you're using in your SOP, then we would say your SOP is not factual. Concise, we mean if there's a way that you can write it in a very short way, please write it in that very short way. But again, sometimes the danger of summarizing things is that they lose clarity. So even as you are summarizing, make sure that it is clear enough. Next slide. Okay, so examples of a poor SOP statement is the sample should typically be centrifuged, then the supernatant may be carefully removed. And believe you me, I've seen SOPs that are written this way. An example of a good SOP of the same statement would be number 1, step 1, place the sample tube in the centrifuge. Centrifuge at 3000 revolutions per minute for 10 minutes. Carefully remove the tube using a pipette, transfer the supernatant to a clean tube. Now we would have explained what you needed to be done. And then there are also languages that we may look out for. These languages may seem like they do not have meaning to us in the lab, but these are the words that assessors and people that have knowledge in QMS look at. So one of them is must. If you use the word must in an SOP, then that is mandatory. If you say in an SOP that staff must wear smart wear from Monday to Thursday, we shouldn't find anyone in the lab casual on Thursday because the SOP says must and it's mandatory. Should we say should in our SOP, it means it's conditional. One can do it and one cannot do it, but they need to justify why they are either doing it or not doing it. Then may is clear optional. Here you don't even need to justify. Okay. So if we do not have technical SOPs in our labs, here is where we begin. For every test that you are doing in your laboratory, it should have an SOP. It means there is already a need for you to develop one. So to begin to develop one, you need to define the scope. The scope means what is the SOP covering? Is it RPR? What is the RPR test going to be used for? That becomes the purpose of that particular SOP. So we have various tests that are used for various purposes. It might be one test. I'll give an example of HB electrophoresis. One lab can use it to diagnose sickle cell disease. Another lab can use it solely for monitoring treatment in sickle cell disease. So as a lab, you need to define really what is the purpose of that SOP? What are you using HB electrophoresis for within your lab? Once you've defined the scope, which is HB electrophoresis, the purpose being either screening for sickle cell or monitoring treatment or both, then now you can assign a person that is familiar with that particular test. That person will be responsible for gathering information. That information will include manufacturer documents, textbooks if necessary, published work, national SOPs, and all of those. They'll get them together and begin to draft an SOP. But we need to be careful not to just copy and paste without adaptation, because we know that some of those things may not apply to our lab. As you are beginning to draft your SOP, you already need to have a template. So in those management SOPs, the link that I showed earlier, there's also a template for technical SOPs there. So you need to have a template where you are now going to be using to feed in information from these sources that you have gathered. Once it's done, you now need to give it to another competent person to look at it to see if it has all the information that is from the source that you were using. Once that is done, you can now assign a document number and version, all of those document control features, and then submit it for approval. Approval now, in this case, will mean that that SOP is number one, adequate and suitable for the defined scope and purpose. So the person that will be approving will now be saying this particular SOP is good enough to use for our scope and for the purpose that we want to use it for. Just to summarize some of the key areas that are going to be in that SOP, number one is the title. So every SOP needs to have a title, and that title needs to be specific. What do we mean by specific? I'll give an example of a pregnancy test. So you'd find this lab is using a pregnancy test that is produced by a manufacturer called Accurate. Another lab is using a pregnancy test by a manufacturer called TZ Check. Others are using Abbott. Those are different methods that are all called pregnancy tests at the end of the day. But since you're writing an SOP on pregnancy test, you shouldn't just say pregnancy test. Pregnancy test using which method? So if it's a TZ Check test kit that you're using, you need to specify and say pregnancy test using a TZ Check strip, using Abbott, using that. You need to be that specific. If it's full blood count using a pouch, you say using a pouch. If it's malaria, you're using field stains or you're using GIMSA, you need to be specific and say we're using field stain or we are using GIMSA. Then you go on to the purpose. We are using it for the diagnosis of this. We're using it for screening. Then you also explain the science behind that particular SOP. You explain the principle. Who is now responsible for implementing that particular SOP in your lab? Are there any abbreviations that you are going to use, any definitions that you need to clarify? All of those, you put them in. You also specify specimen requirements. By specimen requirements, we'll talk about the volume of that particular sample, what are the transportation conditions, and all of that. We also talk about safety precautions. What are people supposed to do as they are testing in that particular test? Then we also mention the materials, that is the equipment, the reagents, and the consumables. We all list them there. And for reagents and consumables, we also list how we are supposed to store them, at what temperatures, and all of that. Then we also describe, if it's applicable, the processes for quality control, calibration in there. Thereafter, we can now describe the step-by-step procedures on how a result will be produced, to include any maintenance actions that we are supposed to take. So here on maintenance actions, let me just make mention. As you are also doing this, remember that we said you are supposed to have forms to keep objective evidence of what is happening through this process. These maintenance logs that you're going to create, please do not just borrow from your neighboring lab. Use these very manufacturer instructions that are there to develop your own maintenance log. You can ask for it for ideas only, but tailor it to what the manufacturer said should be done on a particular piece of equipment. If you miss one step, it means you are not meeting what the manufacturer said, and the quality of results then becomes questionable. If it involves calculation of results, we also say how it calculates results. We also put our biological reference intervals. These are the ones that were formerly known as reference ranges, but they have since been renamed to biological intervals. Then we also put critical values in there. What should alarm us? If we get an HB of what value before we start panicking and reporting results to clinicians faster. Then it should also clearly state how we are reporting results. Are we using a paper-based system? Are we using an electronic system? Are we reporting negative or non-reactive? All of those should be clear in there. And then how those results are going to be interpreted. And also if there are any limitations and sources of error. Now, everywhere we got that information from needs to come under supporting documents. We list everything there to say, for this, we got it here, for that, we got it here. And then we have an SOP that we can use in the lab. After that SOP has been made, we now need to distribute it. We make it available to our people, because this SOP that we've made is not for the people that made it, it's for the patient. We need to make sure that SOP is used to better results of a patient. So we need to distribute it to the point of use. When we distribute it there, do not assume that people are going to read and follow. We now need to train them, show them how it's done. Once you show them how it's done, you now follow them up and see if they're doing it correctly. That process of following up is what is known as competence assessment. We now see, are they doing what we wrote? And if they're doing it, then we say you are competent to do this particular procedure. Once they are deemed competent, they now need to sign and attest that they have read, they understand, and they know what to do. Then you can implement that particular SOP. And what we've discovered in this QMS is that if people are not being followed, with time they tend to relax. So we need to continually monitor the people that we trained and competence assessed and they passed, they are competent. Are they continually competent in this particular procedure? And then we review all of these processes. Do we need to change anywhere? Then we change as needed. So since we are here talking about certification, accreditation, which are just the icing on the cake of quality management systems, we can also discuss how assessors view our labs. So assessors view our labs from only 2 points: documentation in terms of documents and documentation in terms of records. So they will come to your lab and the first thing that should be there is that your SOP should be there. It should be available. Now, when it's available, they will check who wrote the SOP. Was it approved? Does it have the control features that we are talking about? Does it have a proper title? Does it have a control number? Is it the latest version? All of those things they will consider. Once those things are clear, we'll now look at, are the people trained, and then are the people following the procedure. This part of people following the procedure is not by you telling them that, yes, we do follow the procedure. They will now observe you and see how you are performing what has been written down. And then lastly, they will see, is the practice matching the documentation? That is where the real competence comes in. So when you hear that Kawe Central Hospital, where Gomezani is from, is accredited, it means they have proven competence by showing that they have SOPs which are properly controlled, their people are trained and following SOPs, and their documentation is matching what, and their actions are actually matching what the documentation is saying. We have exceeded the time that we agreed upon. I hope no one stones me when you see me in town tomorrow morning. But thank you so much for your attention.
Yeah, thank you so much, Mr. Namakando. People, let's ask questions. Let's put those questions in the chat box. So we have the question and answer. If you go on more, or just when you drag where it's showing share, more, in between share and more, there is a section which says Q&A. Let's put our questions there. Yeah, so Daniel is asking, both accreditation and certification assures the client that the organization has in place an effective quality or environmental management system. If any anomaly is noticed by the client, is the accreditation body or certification body affected in any way? That's the question from Daniel.
Okay, thank you. So after Karen, we have a question from Mr. Polepole Pastor. It reads, thank you, Wilfred, for an elaborate answer. Now, what is the salient difference between national certification and SLAMT.
Okay. Thank you so much. So the difference really is in the approach that is taken to implement. So the SLAMTA really is to prepare you for accreditation. Certification at times may end at certification. The ministry is also looking into certain labs just being certified and certain labs being accredited. It's just in the approach that these two programs are being implemented. So SLAMTA is going to take you through 3 phases. There's training 1, training 2, and training 3. It's more like a program. Those 3 phases, when you complete them and you've shown that you have improved from 0 star to any noticeable star, then they are seeing that you are improving towards accreditation and you can apply for accreditation and not national certification. But there are certain labs that are going to be put under the national certification program. These are not necessarily going to apply for accreditation. These will be certified labs, and the Ministry of Health will issue certificates of certification through the national guideline. And the phases in which this happens is via a series of 4 mentorships, and then they are assessing you in those mentorships. So really the programs are the same. The only difference is in the package of delivery. One follows through trainings, the other one follows through mentorships.
Okay, the next question is the accreditation done on individual tests being performed in the lab, or what is accredited is the lab unit performing the tests?
Okay. So remember earlier we said that QMS is specific. So competence cannot be shown for the whole scope that you do as a lab. You'd find as a lab, you're doing full blood count, you're doing RPRI, you're doing malaria, you're doing all of those things. So for the test that you are showing and demonstrating competence, they will accredit you in that particular test. So like I also mentioned, you are creating SOP specific to the method you are using. So even the questions, even the accreditation itself is going to be specific to the test, and it will even go further to the method that you are using. So if you're doing full blood count and you have 3 machines, and then they accredit you in full blood count, they will accredit you in the machine that you have shown competence in. I hope that answers the question. So it's not a whole lab unit, it's a test, particularly a method that you have shown competence in.
Okay, thank you, Wilfred. The next question is also from an anonymous participant. If SOPs are available, is it also necessary to have job aids? And they the same?
Okay, interesting. So SOPs and job aids are 2 different things. SOPs are the detailed instructions and job aids are the abbreviated instructions. So it depends on how complex your procedure is. If your procedure is easy, it has 3 or 5 steps, less than 10 steps, and it's easy to follow, there is no need for you to have a job aid. It's not a must that you should have job aids. But if your procedure is complex and you want people to be directed to a specific process as they are doing a test, then you need to just summarize it and put it something that you can post on a wall. Remember that list that we went through of what a procedure should have. That list cannot fit on a wall. So you'd want to post something that is just simple. If it means adding 50 microliters of a sample and 2 drops of buffer, and then wait for 15 minutes, those 3 steps can be put onto a job aid, and then you post it on the wall. And then you need to have both now. Where those 3 steps are coming from is a parent procedure. That procedure needs to be available as well. So you can have a procedure alone. It's not in any harm if you do not have a job aid. But if you have just a job aid and you do not have a procedure, then that's a problem.
Thank you. Our next question is from Mr. Polepole Pascal, and it reads, Are MOH mentors available to facilities for free, or a facility must pay?
Okay. Thank you so much, Mr. Pole, for being active in this meeting. It's really good to see the engagement. So mentors are available from Ministry of Health for free. They're available for free, but what is being advised is that mentors should be supporting labs within the province. So for Lusaka province, we do have mentors that are there. We can share contacts of the QMS focal point person, and then you can liaise on how mentors can assist. Otherwise, mentors should not charge you. No one should come to your lab and say, for us to help you create documents, you need to pay us this much, unless for probably private facilities that are engaging you in different capacity. But for government facilities, mentors are available at no fee.
Thank you, Wilfred. So Manyani is asking, please clarify what a control number is in an SOP.
Okay, you want to take that one, Gomez?
So yes, Wilfred, I can take it. Thank you.
All right.
So a control number, this is simply a number that helps us to identify a document. When we are controlling our documents, we have certain features. I think when you open the link that Wilfred shared earlier on, you'll be able to see some features on a document that is called to be controlled. So you have the document title, the document number. I would give an example. Let's say Serenge has abbreviated document numbers by maybe SDH, Serenge District Hospital, and then it also has a number, let's say for example 01, and that number has to be specific to that particular document. If let's say for example SDH01 is maybe personnel management procedure, we shouldn't be able to find another SDH01 in the lab apart from the personnel management procedure. So that's simply how a document number can be explained. Thank you. Hope I've answered your question, Manyan.
So there's a question again from Mr. Pasco Polepole. In case someone wants to initiate QMS, does the institution consult individual mentors, or is there an organization that coordinates mentors? So yes, there is an organization that controls mentors at Ministry of Health, which is the Quality Assurance Coordinating Unit, led by Madam Mutinta Shisholeka and Patricia Mtaleka Fueta. However, these processes of engaging mentors have been decentralized to provinces. So whenever you wish to start QMS at a particular lab, you can reach out to the QMS focal point persons, and then these can see which mentors to be assigned to your lab. Or it can work where a mentor visits your lab and then they go through this path. Mr. Mtale, please go ahead.
Thanks, Wilfred. That's if you are done with your explanation to Pasco's question. So maybe just a top up on that. So the control or the coordination of the mentors at MOH goes so far as laboratories in the Ministry of Health and in government are concerned, plus maybe partner labs, if I can call them that way. But if a lab is like completely private, for example, and then they want to use a mentor, there's also nothing that prevents them rather from engaging with the mentor directly as a consultant, who then works with that lab because it's a private arrangement, especially for private labs. I know some mentors have done that before. And also personally, I encourage that because it helps the mentor to also improve their economic status out of their expertise. So where reasonable, where possible, it's a completely like private institution. There's also nothing that prevents the lab from engaging the mentor directly as a consultant. And I know that some mentors have, as long as you manage the time at your facility as well as your engagement at MOH, they have done that in their private capacity as consultants. I brought this up because that's one route that I have personally used to grow myself, apart from contributing so much to the Ministry of Health. But I was also intentional on how I could develop this consultancy line as well, as a career person, as a QMS expert. So within Zambia, but also even at the continental level, whether it's developing training content or delivering that content, or developing these things that these guys have been presenting about in the meeting. So I just wanted us to also expand our view that that's how we grow our profession, and that's how we also grow as individuals. However, like Wilfred said, depending on the type of facility that's looking for that, you may have to ask MOH whether they draft you into a formal mentor program, or it's a private arrangement that you can make with that facility. Thank you.
Thank you so much for that clarification, Mr. Mtade.
So Wilfred, I've seen one question that is persistent. I would want to answer it. We have it from 3 individuals.
Okay. You can take it. The one about different brands in the lab, if we need to have different SOPs for the brands.
Yes, that's the one. You can take it, please.
Okay. All right. Really, there's no rule whether you should put them in one SOP or you should put them as separate SOP in SOPs. But in my experience, I believe it's easier when they are separate SOPs. So it is specific and you are able to competence assess people individually based on the brand. Should you decide to put them in one, then it means when there's a change in one brand in the instructions, but there's no change in the other, you still need to revise the whole SOP and retrain people in the whole SOP. So really, it should be, especially for labs that are able to control what brands are coming in, especially for those that we procure ourselves. Let's ensure that we work with our procurement teams to ensure that we are buying the same brand every time. My preference is that you create separate SOPs. There's no rule, you can keep them together, but you need to specify in that particular scope that this is covering this brand, that brand, and that brand. And as you are now doing your SOP, you should be able to clearly see that from here to here, these steps belong to this brand. From here to here, these steps belong to this brand. And also in your worksheets, as you are creating those results, we should be able to see which brand was used for traceability purposes. Okay, I do not know if we still have some burning questions, but for the sake of time, we'll need to end the meeting here. A post-test along with the course evaluation, the training evaluation is going to be sent to the email. We ask that we take it promptly, probably between now and Christmas. Individual results of how you performed both in the pre and the post will also be shared. Gomez, do you have any last words before we invite our sponsor for today to give us his closing remarks?
Thank you so much, Wilfred. I have nothing much to say, but just to thank everyone who spared their time to attend this training, as well as PHO, Dr. Nomboende, as well as our sponsor, Siders. Thank you so much, Mr. Mwanga. And thank you so much to everyone that has attended, and we wish to follow up with whatever we've trained ourselves in. Please feel free to reach out if you have any questions, concerns, or if you seek clarity in one way or the other. We are always here to help out. Thank you so much.
Thank you, Gomez. Mr. Chibu, any words?
Thank you so much. It has been a very fruitful and wonderful interaction with people from different parts of Central Province and the country at large. Yeah, this is just one of the kind. We've just started, so we expect more engagement with people from peripheral laboratories. And let's expect more. Like I have said, this is just the beginning. Will also be having even biosafety and biosecurity trainings of the same kind. Thank you, everyone. And thank you for the team for coordinating this. And thank you, Siders, for coming through. You've always been very helpful in terms of laboratory services and biosafety and biosecurity. We are saying that the ministry now is making its own strides with the help of the local mentors that you've blessed us with. Thank you once more.
Thank you, Mr. Chibu. Mr. Mtale, you can go ahead.
Yeah, thank you very much, Wilfred and everyone. I think it's a big honor and a big surprise, that I was called at all to speak. Otherwise, I was also just joining in to support what you guys are doing. This is really, really impressive. I think, Wilfred, before we even met, you had been bothering me, asking questions many years ago. We would meet maybe on Zoom. I share what I knew, you would ask questions, and to just see you guys now taking the initiative and also bringing others along is great, and that's what will build our country. And as I said, 10 of us can be in leadership positions. So within this team might be QMS managers who work for CIDAS or Africa CDC or ASLM or whatever. So don't despise these small beginnings. As you are creating these networks, exchange numbers, reach out to each other, exchange documents, templates, so that other people see what you have done. As I said, we are one country and we are trying to strengthen the system as a whole. So, great start, and looking forward to joining some more of such engagements. Kudos to the organizers, and thanks everyone who managed to join this meeting. Thanks.
Thank you so much, Mr. Mtale, and thank you for the support that we received today. We shall surely reach your office for more. Ladies and gentlemen, Dr. Grant is not on call with us, so this is where we end the meeting. Like I said, we'll post the post test to the email. We'll also post the evaluation of this training. The link will be posted there. We'll also post the registration for today's meeting there, so that we capture everyone's names that attended this meeting. If there are no further questions or clarifications, we'll close the meeting now. Thank you so much for attending.